today about something called synthetic biology. How many of you have heard the term? Okay, actually pretty good traction, which is great. What that really means is that we engineer biology, but we're doing it in a more methodical way than what has been traditionally being since, say, the 1900s, traditionally been done. But you'll hopefully get a much better feel for it with this talk. And I will try to focus more on the ideas of synthetic biology than all of the details of the method. So the idea behind the field and the reason that it's becoming a phrase is probably the most important take-home message of this talk. And what I first want to convey is that it has a lot of potential. So when you look at emerging technologies, synthetic biology on the World Economic Forum's top 10 was number 2, but actually all of those highlighted in red on this list could be impacted or directly affected by advances in engineering biology as well. So if we want to, for instance, find new and improved ways to make food from the same amount of soil, louder? Okay. From the same amount of soil, make more food for people, that at some point will probably require figuring out a better way to manipulate organisms. If we want to have more personalized medicine, in other words, when you are diagnosed with cancer, you get a treatment very specific to your type of cancer within the context of your body, that again could make use of some of the methods that we're developing in synthetic biology. So even though it's number 2 on this list, it's also kind of inherent in all these others, which makes it quite important as an emerging technology. Okay. So why is it an emerging technology? Why is engineering biology suddenly something that is new? When in theory, even in the 70s, we had recombinant DNA. What's so different now? And the answer is represented on these 2 graphs. I hate to put graphs in, so I'm going to try to explain these pretty carefully because I think they're really important pieces of information. So on the left, you see the ability to both read and write DNA sequences. Right, so to make DNA sequences, so we can make the DNA that encodes all sorts of things in life, in strings, at least not in chromosome form, but in long strings of base pairs. So that's the yellow. We can also read the sequences, so interpreting your own genome, getting the actual sequence in your body. We can do that. That's the graph that's shown, or the line that's shown in red. And you can see that for both of them with time, we're going, and this scale here is actually log scale on the y-axis. So we're going at the same pace as Moore's law. So most people are familiar with Moore's law, how many transistors you can fit on a chip. And that's really quick. Like, technology is advancing in terms of computing power very quickly. And our ability both to read and write DNA is going at that pace or even faster. So that's a tremendous increase in technology at our fingertips, and now we need to know what to do with it. And so just depicted in another way, in case that doesn't convince you all the way, this is the cost to sequence a single person's human genome. The cost a little over a decade ago was somewhere on the order of $100 million. Right? It cost a lot of money. And now you can probably, this is actually a little bit dated, so 2011, I'd say we're down under $10,000 per genome. So that's a lot of information. That's a lot of DNA that we're getting the sequence of in each person. And we can do that, and it's getting faster and cheaper. Still, it hasn't slowed down at all. In fact, it's accelerated. So we have all this information about what's encoding all of our organisms, and we have this ability to make a lot of sequences. And the question is, what do we do with that information, and what do we make, and how do we make it? These are really important questions. So this is a cartoon kind of depicting a video game version, so very artificial, but the idea is that we can recode DNA now to create a new function in an organism. We're not great at this yet, but we can do it. And we can especially do things like make a new function where it used to make ethanol. Maybe we can make an organism, instead of making ethanol, make butanol, which would be a better fuel than ethanol. So we can make incremental changes at least. And doing this is a lot like rewriting some computer code to tweak some things, right? So getting an entirely different function right now is a lot less feasible, but that's where the field would be going. The biggest application space right now for synthetic biology is actually in the production of fuels and chemicals, which is why I used that example a moment ago. We can make, very well, we can make medicines in organisms, right? A lot of small molecule drugs. So in other words, something like the anti-malaria drug artemisinin, which is now, I think, to the point of actually being distributed in Africa at a very low cost, is being produced in yeast. The many, if not all, protein therapeutics that I know of are synthesized in an organism. And so that includes things like Herceptin or Avastin. So there are a lot of drugs that you probably have heard of that are made out of protein, and the protein is in turn made in an organism. And to get organisms to make these things is not usually natural. We usually find a natural source, but we can't scale that up. We can't make a lot of it in that natural host, so we have to move it to something new, and that's what synthetic biology does. Lately, let's say the past 10 years or so, it's been especially important to a lot of synthetic biologists to make an impact on the world that's helpful, and so particularly with respect to the environment, there's been tremendous investment of time and resources into making biofuels. So these are fuels that have been generated, so in some way sunlight is converted into a form of energy that's stored in plants, so biomass, or algae, something that's growing, and we can convert that into a form that we can use, so a liquid fuel for jets or for cars. So biofuels and at the same time biochemicals, because there are a lot of things we get from petroleum that do not include fuels, but that are chemical precursors. So probably your baby's diapers would be made of polymers that are actually made from derivatives of petroleum. And we could replace all of that carbon usage with something from a renewable source if we can just figure out how to make the molecules in organisms. And so we're figuring out how to do that. And then finally, even the precursors to a lot of fabrics. And by fabrics, I mean carpet fibers or particular blends of cotton-like material that form some of the textiles that now, at least in the couture lines, are being incorporated from organisms that have been engineered to produce them. So it's all around us. We don't really think about it that much. What we can do with organisms, with living things, to help them produce exactly what we want, so we don't have to manipulate it so much. So we have this explosion of technology, right? We can read and write DNA sequences. We can recode that DNA for new functions, albeit incrementally different in most cases, and then use them to synthesize chemicals, drugs, and fuels from renewable sources. That's not the main idea, I think. The really key point is where we're going to go with this. Where else can we go? This is what we've been doing. In fact, we've done a lot of these things from the mid-'80s, I'd say. So of course we're going to be able to make more fuels and chemicals. This is what I see right now in the synthetic biology community. A lot of people are coming up with really clever ways to make a variety of different fuels and chemicals from a renewable source, say grass clippings or grasses that grow in marginal land. So that's one. But I also see a lot of work in several other areas that I'm going to highlight. So there's this idea of bioelectronics, interfacing biology with inorganics, so computers. So imagine a wire that's somehow connected to a living organism and either sending a signal down in digital form, because that's usually how the signals are going to be transmitted, and telling the cell to do something either with the on or the off portion, and then the cell has some output depending on what the wire is saying. Or vice versa, maybe a bacteria that's connected to a wire actually depositing electrons. And so you have a living battery, you have some organism that takes in sunlight and directly converts that into electrons that you can use out in the field maybe where there isn't any other power. So this would be directly using the organism for energy rather than going through an intermediate step where you store it and then you have to break it apart and make a new molecule. So bioelectronics. Biomaterials, I've talked a little bit already about how we can make some of the precursors for things like carpet fibers and certain textiles. There are a lot of materials out there that are entirely produced in organisms already, right? Cotton is one of those that actually already come, so we just refine that. Spider silk turns out to be a really interesting material as well. It's stronger than steel, tougher than Teflon or Kevlar, depending on what you want to call it, and very elastic, so you wouldn't want to use it as a bulletproof vest. The bullet would be stopped, but not before it actually went in you with the spider silk fabric. So you pop it back out, but that wouldn't be so great. But you can imagine that might have some utility in the world to have these different materials with such different properties than what we have. And these types of materials turn out to also be made out of protein, which means that we have to produce them biologically. There's not even another way to synthesize them. And so we have to figure out how to get them out of things other than spiders. I don't know if you've ever tried to grow more than one spider at a time. It turns out they're pretty territorial. You can't scale that up. You can't make a factory with a ton of spiders inside and pull their silk. That just doesn't work. And so we need to find some other organism that can make spider silk for us that's more amenable to being grown in small batches that produce a lot. So more far out there ideas that are probably coming in a couple more decades, but the ideas are already here. The idea of altered environment. There's a Kickstarter a year and a half, 2 years ago now, I think, for glowing plants. And the idea is, behind this project, to plant a tree that has something encoded in it that makes it glow at night. So you don't need street lamps anymore. You don't need electricity. You just have the tree planted. It's doing its thing during the day. It's great for the environment. And then at night, it also glows and lights the path. It's a really cool idea. It's feasible to make a plant glow, probably not bright enough to be a street lamp, but the idea is already there, right? So we just have to do it. Of course, that's not natural. Plants don't naturally glow, but there's the firefly enzyme could be moved into plants. So altering our environment, that's one idea. Other ideas I've heard are making a tree, so engineering it a little bit, so instead of growing straight up, it actually grows into the shape of whatever it is that you want to build. Can we start to control the shape of the tree? So, maybe it grows into the shape of a house instead of just straight up and down. Cutting out some steps. Again, we don't have the capability to do that yet, but people are thinking about what would it take to be able to do that? What in the DNA tells the tree to grow in particular shapes with the particular types of branches they have? These are all really interesting questions that come out of just trying to think about engineering these functions. And finally, I already talked a little bit about personalized medicine. We have to learn a lot more about what all of the DNA means. So we can sequence the genome, but we don't actually know what all of those pieces mean. And so figuring out what they mean and how that's going to impact how every single drug is metabolized within somebody, how much more likely you are to get cancer than someone else to begin with. All of these things are questions that we need to answer still. And last but not least, I think the most transformative technology that's going to come from synthetic biology is probably not any of the other categories. It's going to be something completely new that somebody is thinking of right now and developing right now, or maybe they're in classes right now just learning about synthetic biology, but they're going to come up with something that can truly change the world, and hopefully for the better. So what is our challenges? It would be incomplete if I told you all of these possibilities without telling you what the problems is. And it turns out there are a lot of problems, and all of them pretty much have to do with the fact that biology is really complex. There's a lot of DNA in a human, for instance, right? And all these functions that it encodes interact in ways that we don't completely understand, and we need to understand that if we're going to, one, manipulate it, or two, have a chance at doing so in a way that's actually towards the goal that we want. So one of the ways that biology is complex, I'm just going to highlight a couple there. There are so many, I just want to give you an idea. But one of the ways biology is complex is that organisms respond to changes in their environment. So it's not like a test tube. So I think a lot of people that engineer biology like to think of it as and A goes to B reaction, but as soon as you add A, the situation has changed for the organism, and so it won't necessarily just go to B. It might go to B under one particular circumstance, but then on a different day it doesn't, because maybe the pH is slightly different, or the sunlight is at a different level, right? So all of these factors from the environment actually change the response that we get. Within the cell, things can change a lot. There's simple systems where you have some sort of input leads to some sort of output, and that's directly regulated by one component of the cell. That we can figure out when that's the way it works, right? Because we can eliminate that component and we eliminate our output. But often in cells, they're regulated by something much more complex, where maybe there's 3 different components that are sensing the same input, and the way they react to that input dictates the output of the response. That's actually the way the human nose works, right? We don't actually have a sensor in our nose for every single chemical or molecule out there. We have just a few, and it's the pattern that your nose nerves actually detect. It's interpreted by your brain to be a particular smell, right? So the same is true for most of what's going on in cells. And it's great right now. We are really adept at working with simple systems, and we're just starting to learn how to work with the complex systems. Another way it's really complicated is that we need to develop new ways to organize activities inside cells. So right now, a lot of engineering biology resorts to putting new pieces of DNA in and treating a bacteria, so just a microscopic organism, as a sack for holding that DNA and turning it into enzymes. So it's just sacks, little factories producing what we want from A to B. But in fact, in nature, that's not how it works. There's usually scaffolds for all the pieces to make component A turn into component B. And so we're going to need to learn how to organize all of the activities that we want to put into cells, or else it's never going to produce all that we can dream of. There's 2 ways of doing the scaffolds. One is to kind of line up like an assembly line. Another is to compartmentalize. And I actually work in my lab. I won't talk too much about my own work today, but we spend a lot of time trying to figure out how to introduce new compartments, entire compartments into cells to house the artificial reactions that we're introducing. And so that's kind of inside a cell, how you might need to organize, but you also need to organize cells within a network of other organisms, right? Usually things are not isolated in a test tube. In fact, this right here is depicting E. coli that, so the center spot was placed on a petri dish and they were allowed to grow outward. The center spot is just well mixed with E. coli that either had a green fluorescing protein or a red fluorescing protein. And they formed this pattern spontaneously. But if they do the same thing in yeast, so Saccharomyces cerevisiae, we get a different pattern. But it's just a well-mixed set of organisms. So they differentiate into patterns on their own in some way. And those patterns are different. We don't understand this yet, but we need to understand how these organisms are interacting. And then finally, we need to figure out how to get material and information in and out of cells. And this is the bulk of the work that goes on in my lab, actually, is figuring out how material and information are flowing. Primarily, we look right now out of cells. So this is a cartoon of a gram-negative bacteria. So it has a very characteristic shape. These are the flagella that help kind of move it around. The DNA in the middle, and these are the membranes that are blown up here. They're called phospholipid bilayer membranes. So if you remember anything from a early high school biology, that's probably what it would look like in the textbook. And that membrane helps protect the cell, so keeping toxic things out, helps facilitate the crossing in for sugars and other food type substrates, and the efflux, so the transport out of waste products. And it does this all just with a single membrane. What we have at the macro scale is a variety of different membranes and wires and all sorts for all these different molecules. The cell has a single membrane, it does all these jobs. So it's pretty incredible. And we can use, if we could control this, we can use cells much more efficiently to make chemicals, because we need to control what's coming in and what's going out in order to make chemicals more efficiently. We could also potentially control photosynthesis mimics, if we can control electron flow into and out of the cells, I mentioned with the bioelectronics example earlier. Or we can even get to detection of toxins in the environment or in the body through sensing by using information flow into and out of the cells. So this idea of moving things across cells is very important, but it turns out that the cartoon that's in a lot of the textbooks is not totally accurate. There's actually a lot of blobs sitting in that membrane, and those are proteins as well. And those proteins are forming pores and transporters for all these different molecules, electrons, and so forth. And so that's what we actually have to engineer. And there's two ways to do that. We can change the transporters that are in there, find the one that does what we want, and put it into a new organism, or we can change the number of them. So if we want to modulate how much is going in and out, we can, let's say if we want to increase it, put more transporters in place. So that's sort of the idea behind a lot of the work that goes on in my lab. What that's led to is we engineered an antibiotic pump to, instead of pumping out antibiotics, which is its natural job for a bacterium that lives in the gut, that's a very sensible transporter to have, something that secretes antibiotics and makes it resistant. But we've actually changed it so that it acts on butanol instead. So we changed the specificity for this transporter so it can pump out the fuel, which is otherwise toxic to it. So if it pumps out the fuel, it can make more of that fuel because it will take a lot longer to reach a toxic level inside the cell. And that actually works when we're trying to produce fuels. That's what this graph shows, is that when we engineer the pump, we get a lot higher, tighter butanol. Sorry. Is that my timer?
Yes, your timer.
Yeah, okay. That is fine. I had one more example, which I don't have to go through in great detail, but we actually engineered a spider silk to have a, so it's actually not exactly spider silk, it's a different material protein, but very similar, to have an antimicrobial peptide from a frog skin on it. And that material that we made was naturally antimicrobial, so when we didn't have that peptide present, the cells were alive still, they're green, and when we had it present, they were red, so it was killing the salmonella just by being present in our engineered material. So in summary, I think we have to learn to control these complex biological or bio-inspired systems, and there are a lot of ways in which we can do that, but there's a lot of challenges for anyone that wants to tackle them in terms of understanding what's going in and out of cells, but also all of those other challenges I mentioned. So scaffolding for what's going on in cells, learning how they interact with one another, learning how all cells respond to all the inputs that it's receiving, and so forth. So that, I will keep up a picture of my group. That's my website. If you want to learn a lot more detail about all of the projects going on in my lab, it's at that website, and I can take